Hi, my name is Peter Milner. I'm a basic scientist and a physician, trained at Liverpool Medical School in the 1970s and graduated with honors in medicine and biochemistry in 1980. I then did an internship in Liverpool, then moved to Johns Hopkins Hospital to do a medical residency and study genetics from 1981 to 84. I was boarded in internal medicine in the United States, and then continued my education down at the University of Virginia in Charlottesville, where I studied cardiology for 2 years and was boarded in cardiology and became a fellow of the American College of Cardiology. I also did research with the Department of Pharmacology at the University of Virginia, Dr. Michael Peach and Dr. Bob Byrne, and during that time showed that endothelium-dependent relaxation, which was a newly discovered factor, was actually not a arachidonic acid metabolite. And we published that in the Journal of Clinical Investigation. I collaborated with Sir John Vane, who was a Nobel laureate and became a good friend. He had discovered prostaglandins. After the University of Virginia in 1986, I was recruited by the Hematology and Oncology Division at Washington University in St. Louis. They were prominent basic scientists in medicine, Dr. David Kipnis, Dr. Philip Majaris, Dr. Stuart Kornfeld, and Dr. Tom Dool. So I set up a lab there and worked with Dr. Tom Dool, who was my mentor, on platelet-derived growth factor, having shown that he showed that it was a homolog of the V6 oncogene. He then went on to study to show that it was involved with wound healing with Dr. Tom Musto, who was a plastic surgeon, and Jean Bauer, who was a dermatologist. Out of that came a successful product with Johnson & Johnson Cell for wound healing. Meanwhile, in my lab, I was looking primarily at angiogenic growth factors, and I identified a novel angiogenic growth factor in bovine uterus that was responsible for vascular growth in the endometrium. I was able over 2 years to purify a sufficient amount, microgram quantity of that protein to homogeneity, and working with Dr. Ned Siegel at Monsanto, we were able to get novel N-terminal sequence. And with that sequence, we were able to clone and sequence the cDNA and publish the protein and the discovery of the protein in the sequence in Science and call the protein pleiotropin. I then went on to look at the gene and cloned and identified the gene and chromosomally localized the gene for pleiotropin. And showed that it was regulated by estrogens and progesterone and by retinoids and involved in basically embryo growth and differentiation. Another group in Finland showed that, or simultaneously discovered the protein pleiotropin as a neurite outgrowth factor in the brain. Since then, a tremendous amount of work has been done on pleiotropin and its receptor RTP beta zeta, which activate a phosphatase or inactivate a phosphatase, which is the phosphatase for ALK kinase, anaplastic leukemia kinase. So that whole pathway of pleiotropin and its related protein midkine are now important growth factors. I then went on and left WashU in 1992 to move to Stanford, and then soon after founded a company called CV Therapeutics, where I in-licensed ranolazine with my co-founder Lou Lang, and then invented regadenoson, which is an important vascular imaging agent. That company, CVT, was sold to Gilead for $1.5 billion in 2009. In 2000, I left CVT and founded another company, Arix Therapeutics, dedicated to soft chemistry, where I invented and developed with the rest of my team 3 drugs, budeodrone, tecarfarin, and neuronopride, and raised about $150 million. Those drugs are in late-stage development. The 2 cardiovascular drugs are with a company called Aspero in the United States, and neuronopride, the GI drug, is in late stage development in China with a company called Sinovac. So I work closely with Chinese drug developers who are dedicated to developing drugs globally based in China. I'm also served now as the Chief Medical Officer and board member of Retrotope, and we have drugs for ALS, infantile neurocternal dystrophy, and Friedreich's ataxia that we're developing in the United States and looking at developing in China. Thank you.